Well, welcome everybody. Unfortunately, I'm sharing screen and I can't see all your faces, but welcome and thanks for joining us for Beets, Breeding for Flavor and Color. We're just going to get started with some just housekeeping and information about the interpretation. So Siri, if you could just read this slide. So people will see that there's
Okay.
Yeah.
So for the webinar this year, we are providing simultaneous translation to allow participants to listen to the language of their choice. You can do this by selecting the language interpretation option in the settings bar to mute the original and hear the translation as shown on the screen. Alors cette année, nous offrons la traduction simultanée.
Now what you're getting is the instructions in French for getting the simultaneous interpretation. So this is a good test. If you're hearing my voice, then that means you found the English simultaneous translation function.
Okay, thanks. So I'm just going to quickly say a land acknowledgement. Thanks for coming here from wherever you are.
Merci beaucoup d'être venu de là où
While
vous êtes.
we're meeting virtually today, I'd like to take a moment to acknowledge the importance of the lands which we each call home. The land we live on, work with, and plant our seeds into. From coast to coast to coast, we acknowledge the ancestral and unceded territory of all Indigenous people that call this land home. To recognize the long-standing relationship between the land and all the Indigenous people, which precedes the establishment of Canada and is affected by the violence of colonialism, capitalism, and ongoing racism. By living here, working here, and using the resource of this place, we now participate in this experiment. And as part of the process, as part of the process to be accountable to our role on this ongoing violence, we invite you to reflect on these questions with us. What are our obligations to the Indigenous people who first inhabited and continue to inhabit this land? How can we support existing local struggles of Indigenous people along with preservation, resurgence, and development? And how do we live and farm as good treaty people? We also encourage you to learn the history of the land you're in relationship with by visiting www.whos.land and native-land.ca. In working through this webinar and communicating together, we would like to cultivate a safer space. We aim to create a supportive and non-threatening environment for all. We hope presenters will feel valued and respected and may freely choose not to answer a question. And we hope participants will feel comfortable asking questions because it's the best way to learn together. If you tend to speak a lot, we invite you to move up and listen a bit more. And if you tend not to speak as much, but you have burning questions, we invite you to move up and try to ask them. And we speak from the I and define the we. If you want to engage that with questions or comments, first of all, please make sure your video is off, but if you'd like to, or your audio is off, but if you'd like to ask a question at some point, you're welcome to turn that on and ask a question, or you can add it to the chat box below. If you can make sure you stay muted while not speaking, that's great. Just introduce yourself in the chat and ask your question. And without further ado, I think we will get started. So I'm going to stop sharing my screen in a second and just introduce Solveig Hansen, who is she's a postdoctoral research fellow at the University of British Columbia and overseeing the Canovi trials, and has done a lot of work in beet breeding and participatory breeding as a farmer and with farmers. So without further ado, Solveig, feel free to share your screen.
Okay, can you all see that?
Mm-hmm.
Can you see the notes version or the big picture?
Big
picture. Great, that's what we want. Okay, like David said, my name is Silvia Hansen. I'm excited to be with you here today to talk about one of my favorite crops. One moment, gonna minimize, there we go. Okay, I finished my doctorate in plant breeding and genetics with a minor in life science communication at the University of Wisconsin in Madison this past December, and then came right here to start this position in January, working with the CNOVI, Canadian Organic Vegetable Improvement Project. My doctoral work focused on beet flavor, specifically geosmin, which is the compound that confers earthy flavor to table beet. So as part of that work, I led a participatory beet breeding project in and for Wisconsin organic farms that resulted in some novel varieties and also some insights into the relationship between flavor perception in beet and hedonic liking, or just overall liking of beet. And as I was doing this, I also just fell in love with the beauty of beets. I was excited to see beet breeding and beet seed production happening here in BC. So I'm excited to tell you the story of this particular beet breeding project, tell you what we learned about beet flavor, color, and how flavor and color interact, and then think with you about ways to evaluate, breed for, or just appreciate the color and flavor in the beets that you're growing and breeding here. There we go. Okay, to help you place me in space and time, I grew up in Southwest Wisconsin in the United States. I was an outdoor kid. That's right. I went to college. I caught the organic farming bug, and then I spent 8 seasons as co-owner of a direct market vegetable farm in Iowa. I found the seed world when I took a customer care job at a seed company in Western New York, which turned into an organic product manager position. As a farmer, I had become very aware of the differences among varieties. But as a product manager, I got to observe them systematically. I still, however, didn't know how they came about. And that's when I serendipitously found the Student Organic Seed Symposium in 2012, which introduced these concepts of plant reproductive biology, population creation, selection. And I realized there was a chance I could learn this. So at age 35, I went back to graduate school at University of Wisconsin-Madison, which was serendipitously close to home, and started my prerequisite classes. I share this not only to illustrate the way professional and personal lives co-develop, but to acknowledge that my story is shaped by several privileged identities, especially my identity as a white settler with upper middle class socioeconomic status. Those identities have shaped my emotional and intellectual landscape, my ability to make money and fail to make money, the questions I ask with both my research and my life and this presentation I'm giving you right now. All of the research I will present today was conducted on the ancestral lands of the Ho-Chunk, Menominee, and Oceti-Sakowee nations in what's now known as central and southern Wisconsin. And I'm zooming to you today from the unceded ancestral territories of the Squamish, Musqueam, and Tsleil-Waututh people. There we go. After finding plant breeding, I found beets, and I found them through a person. That person was Dr. Erwin Goldman, who invited me to join his carrot, beet, and onion breeding lab at Wisconsin. Beets were appealing to me because they are widely grown in Wisconsin, both for processing, as in the giant mountain of beets in front of which I am dressed like a beet, but also for organic growers. And beets, in addition, are having a bit of a moment. Beet was brought to the Americas by European settlers, but until recently it was considered a simple food, a humble food. Lately, though, beet has become iconic of farm-to-table culture, so I thought it held particular potential for engaging local produce consumers in Wisconsin. Another reason beets are fun to work with is that people have opinions on beets. It's generally agreed that sweet, earthy, bitter, and harsh flavor attributes are prominent in beet, And each one of those is associated with one or more flavor compounds that are listed there. The earthy flavor is the one that's most commonly linked to preference or non-preference for beet. Some people say beets are dirt candy. Some people say beets just taste like dirt. Importantly, perceived flavor doesn't just depend on the chemical compounds you put in your mouth. It also depends on the specific assortment of flavor receptors each one of us has in our mouth, and also our brains, the emotional and cognitive environment in which those flavor stimuli are interpreted. And those all vary among individuals. So two people eating the same food can really honestly taste different things. That notwithstanding, we know that flavor is of particular importance to consumers who purchase directly from farmers and those who buy organic food. So it's important to know whether the flavor compounds we measure, like geosmin and sugar, relate to perceived flavor, and especially flavor liking, how much you want to eat a thing again. The earthy aroma in beet is conferred by geosmin, which is this molecule right here. It's a volatile molecule, meaning it's aromatic. It is known to be synthesized by a wide variety of bacteria and fungi in the soil. It is the scent of freshly turned earth. Humans are very sensitive to it. Historically, it was assumed that geosmin was in beet because beets grow in the soil, so that they were absorbing this earthy aroma from microbes that lived in the soil or microbes that lived like within the beet tissue. However, selection or analysis of beet tissue grown in sterile tissue culture and also in heat sterilized soil still showed that beets produce geosmin in cultivar specific levels. And even adding to this, we find that geosmin can be selected for. So this is a bidirectional breeding experiment where people selected for high geosmin over 3 seasons and low geosmin over 3 seasons, and saw that it is heritable. So we see that geosmin appears to be produced by beet plants. It's within the beet genome instead of being absorbed from the environment, and it's heritable. And that means we can make new beets or beets with defined levels of geosmin, which could give new flavor experiences. And in particular, if geosmin or that earthy aroma is a reason that some people don't like beets, beeting breeds with very low geosmin could open up new market space for beet, new possibilities for this vegetable. Serendipitously, this project, this bidirectional breeding project that my predecessor, Lynn Mayer, did as part of her doctoral work, yielded these 2 populations with very high and very low geosmin, segregating for a brilliant array of colors each one, which made them perfect for participatory breeding. They're well suited for direct markets where variability and novelty is valued instead of a detriment. So I use this participatory plant breeding approach for a couple of reasons. One is that I wanted to end up with varieties that really worked on organic farms and that were really accepted and liked by those farms' consumers. So participatory plant breeding, as many of you likely know, was first developed in developing countries for farmers growing usually grain crops on marginal lands that were very different from the conventional breeding environments where those crops were bred. In wealthier countries like Canada and the US, participatory plant breeding is used again for growing environments like organic farms that are very different from conventional breeding environments. So we're selecting in the target environment with the target bunch of consumers, with the actual people who are going to be eating these things. And the other reason I did this is because I like working with people. It makes me happy. And core principles of the organic movement are stakeholder engagement, open knowledge exchange, and just community building. So the 2 goals of this project were to develop consumer accepted specialty beet cultivars, and then also use those cultivars to investigate whether people could perceive and whether they liked beets with these extreme levels of geosmin, more or less than were available in the marketplace before. So I used two models. One I'm going to refer to as the farm model, and that was conducted all on this farm down here, TP Produce in Evansville, Wisconsin. So for that model, the people who selected were the farmers on that farm, one of whom is Beth Kasmar, tasting a beet there, their staff at one of our staff tastings, and then their CSA members. On the other program, which I called the outreach model, I surveyed farmers, so farmers from many farms. I had a group of chefs who served as the core group of tasters, and then I went to a tasting event to reach consumers who bought from all sorts of different farms. This was the selection cycle for the first 2 years of participatory selection. So say we're talking about low geosmin beets here. I would have 30 families of low geosmin beets. A halfciv family in beet, as many of you likely know, is all of the seed from a single plant. So it's one seed parent who contributes one gamete to each seed, and then lots and lots of pollen parents that wind pollinate and land on that seed parent. So 30 of those halfciv families, you'll see that a certain number of families are eliminated at each step of selection, and I'll go through that process, leaving us each year with 5 families that then are intermated to create the next year's starting population. Plant breeding requires selection from a variable population. And this plot, this is one of the few, like, you know, statistical plots I'm going to show you today, shows very clearly that the geosmin levels of this population, or these two populations, are very different. So we see the big difference in mean geosmin between the two populations. We also see a difference in variability. So geosmin is a compound that increases in its variability as the concentration itself increases. And as a result, we saw significant differences among these low geosmin families, where we didn't see significant differences among these high geosmin families, even though the means were still all bouncing around. So that foreshadows that there might be more possibility to move the mean in low geosmin beets than in high geosmin beets, just because there's so little variability within a family. For soluble solids or sugars, we don't see much difference at all, which makes sense because these populations weren't selected for sugar. So we see a lot of variability and quite a lot of variation within families. First, we did horticultural selection, which is where we asked the farmers what was important to them in beets that they grew on their farm. So I gave them this survey and asked them to rate different characteristics. In both models, the farmers indicated that high germination, ability to outcompete weeds, and disease resistance were top priorities. That probably doesn't surprise any of you who grow anything in organic environments. Then with the help of my lab team, I harvested 5 roots from each family and took core samples like this for geosmin analysis and sugar analysis. And then we also took all sorts of phenotypic variables. Did a quantitative selection using a selection index, which I can tell you about later if you want more details. I also made qualitative comments about each family because, as a lot of you know who have done breeding, once you look at a certain number of families of a crop, you kind of just know, like that's a good strong one, and that one it either is just too variable or just doesn't look healthy. So using those methods, about a third of the families were eliminated. So we got down to 20 halfciv families. Then I sorted them using color into groups that gave the core selectors something to choose from. So then we went to hedonic selection, which is the fancy way to say how much do you like this or how much do you want to eat this again. We started with a core group of tasters in each model. So on the farm staff served as that first group of tasters. For the outreach model, there's a group of chefs that has been gathered in Madison, Wisconsin by Dr. Julie Dawson, called the Seed to Kitchen Collaborative. So they did the first round of selection, narrowing it down from 4 high geosmin groups, 4 low geosmin groups, to 2 of each. And then the consumers did the last round of selection. So they chose 1 high geosmin group, 1 low geosmin group, and then those 5 families were intermated to make the starting population. Ooh, there's more I wanted to tell you about that. In the majority of those tasting instances, there was no significant preference for the selected group. That could be due to the fact that there were no significant differences in sugar between any of the groups. There were 2 cases in which geosmin was different among the groups presented to consumers, but neither of those cases were associated with a significant difference in choice. We also found that taken together, there was no significant difference in liking between all the low geosmin groups and all the high geosmin groups. So we didn't see any systematic preference for geosmin in these steamed beets, despite the fact that earthy flavor is widely reported to be associated with liking or not liking of beets. We did find 2 big disparities in beet liking and in appearance liking. And each of those came from a comparison of a white beet group and a pigmented one, on the order of 25% liking for the white group, 80% liking for the pigmented group. But in both cases, liking after the people ate it was roughly equal. So the flavor seemed to be positive enough to offset that significantly less liked appearance. We know that in other foods, low color is often associated with an expectation of low flavor. So if that occurred here and people weren't expecting much from these white beets, the technical term that we observed would be positive disconfirmation of expectations. But that just translates into getting more than you thought you would, which is something that people generally like. Notably, both of these white beet groups that produce this phenomenon of low appearance liking and high overall liking were selected by chefs. And chefs are, culinary professionals who both create cuisine and create expectations around that cuisine. So it's very interesting that they played this central role in selecting beet groups that had this very different reaction than other groups. Oh, there's my friend Dan with a delicious beet brownie and white beet sorbet dessert. So in this case, we selected on overall liking, just how much do you like these things? But then we measured on the back end, how did geosmin move? And just like we predicted, the low geosmin population did diverge. So this is the 2 cultivars that came out of the project were significantly different in geosmin concentration. Didn't happen in the high geosmin population, likely because those families were so variable. We could pinpoint the 2 selection events where a group with higher or lower geosmin was selected, but those weren't so associated with differences in people's votes for the families or for the groups. So this divergence is really due to genetic drift, or just the phenomenon of kind of a genetic bottleneck, in this case, artificially created through our selection process. So here's what we came up with. For the low geosmin selections, the population started out as segregating for the 3 major colors in beet. I can tell you more about that biochemically later if you're interested. But you'll see that the outreach group, driven by the chef's preferences, selected a white low geosmin beet. The farm group selected a traditional red low geosmin beet. It's very mild. This Evansville Ember is actually going to be commercialized next season pending a strong seed production by Fruition Seeds, which is out in Naples, New York. So it's really exciting that that one will actually be growable in gardens and farms soon. But notably, these geosmin levels are low, but there are plenty of cultivars on the market that have similar geosmin levels. So the geosmin doesn't give a different flavor experience necessarily than other beets you would find. The high geosmin selections are a different story. The geosmin concentration in these is roughly double that of bull's blood, which is the highest geosmin commercialized cultivar that we've been able to find. So these do represent a new assortment of flavor compounds. The outreach side of the program selected 2 varieties. Actually, moving target, this zone beet with some coral colors actually won the consumer selection, but this white one was a narrow second, and I thought it looked so unique with the pink petioles that I couldn't help it, and we just kept it anyway. And this one, the Blushing Not Bashful, is also going to be commercialized by Fruition next year if all goes well. The farm group also chose a zoned beet, but with relatively less pigment, so it's closer in color palette to the Chioggia beet that many of us know and love. The moving target has a lot more pigment and more of these coral tones. After those 2 cycles of selection, one moment, okay, I think I'm doing okay on time. After these 2 cycles of selection, we had selected for the visual phenotypes. We knew what colors we were going for. We knew the root characteristics we wanted, the top characteristics we wanted. So we asked the chef and consumer participants to turn their attention to flavor evaluation. So what did we get out of this? How can we describe it? I will, at the very end, I'll describe how we made this ballot, but basically we used a ballot using some of the salient flavors in beet, so sweetness, earthiness, bitterness, and just asked people what they tasted in these newly developed cultivars alongside some checks that were more familiar. This was done over 2 years. Okay, for sugar, or for perceived sweetness, which is different than sugar, perceived sweetness varied significantly with both cultivar and cultivar by event interaction. But some cultivars were perceived as consistently more sweet than others. So Blushing Not Bashful, which I indicated with this pink burst, that was in the group with highest perceived sweetness in all of the tasting events. Snow Globe with the snowflake was in the highest group in 4 of the tasting events. But interestingly, there were no differences in insoluble solids or sucrose among these cultivars, despite these differences in perceived sweetness, which is interesting, but this has also been observed in other crops where bitter and volatile aromatic compounds can mask or enhance sweetness. So perceived sweetness has to do more, it's not just a function of how much sugar there is, it's a function of how that sugar interacts with the other compounds in the beet. So this could be happening in beet. So the overall flavor is a function of interacting flavor compounds, not just sugar. Earthiness, perceived earthiness also varied significantly with cultivar in both years. But in contrast, no varieties were found as systematically more or less earthy. That is despite the fact that there were very significant differences in geosmin between cultivars. So in all these places, I've put the red boxes. The black bars are the high geosmin cultivars, while the light gray bar is Snow Globe. That's the low geosmin white. So there are 2 to 3 times as much geosmin in the black, the high geosmin cultivars than the low, but we didn't see a significant difference in earthiness perceived in any case. However, significant differences in earthiness were perceived between cultivars with no difference in geosmin. So this kind of adds to our evidence that there's more to earthy flavor than geosmin concentration, or maybe that the word earthy isn't descriptive enough to get to the difference in flavor experience that people are getting from beets with different geosmin concentration. So differences in earthiness were not associated with geosmin, but they were associated with differences in sweetness and bitterness. So earthiness was negatively correlated with bitterness. Wait, nope, this box, yep. And positively correlated with earthiness, which basically means people might not actually be tasting something separate, something they actually perceive as earthy, but they could be interpreting the word earthy as high in bitter and low in sweet. We also see that flavor liking, maybe not surprisingly, is positively correlated with sweetness, negatively correlated with bitterness, but it's not consistently correlated with earthiness. So we find that earthiness, perceived earthiness, was not consistently associated with either geosmin or overall liking. So what does this tell us? It tells us that geosmin concentration is not the central determinant of liking in table beet. It's not the one element that that liking is controlled by, even though people talk about beets tasting like dirt as a reason they don't like them or do like them. We learned that a broad range of geosmin concentration can be well liked. We learned that manipulating expectation, like with color, could be as effective in manipulating flavor perception as manipulating the actual flavor compounds. And we learned about the possibility that flavor compounds could interact to produce the flavor sensation that we experience. We also learned that our vocabulary might not be rich enough to describe the differences that might be actually perceptible in beets with different geosmin levels. So this lexicon development project is something that I started but didn't get to finish. Real briefly, I had 10 chefs, 8 varieties of beets, like you see right here, and a blank sheet of paper. And I said, what does this remind you of? Like, just brainstorm words on, for taste, aromas, and textures that these samples remind you of. We got 270 words in about 20 minutes. We sorted them, classified them, worked with a small focus group of chefs to eliminate ones that weren't as good. And it's from that I got the words from the ballot that you saw. But you know what, I'm going to flip right to here. Can I? Come back. There we go. But we used these words that didn't go on the ballot, which were most of them, to make this flavor wheel. This is just a draft, but this is our best working guess at different categories or different flavor descriptors that might help to describe beet. In the trials I did with this, I gave people both this ballot, like I showed you before, and the flavor wheel. And I'm curious whether the flavor wheel helps to differentiate between any varieties that are not differentiable with just the ballot. So if any of you would like to use this flavor wheel in anything you're doing, help me refine it, take out any words that just don't make sense to people who aren't chefs and haven't spent hours and hours tasting wine and coffee and developing a flavor vocabulary. It would be really cool to develop a functional flavor lexicon that can be used by farmers and breeders, I think. So now we turn to the work that you're doing. And so I'll just put out a few things for you to consider as you're tasting the beets you're growing, as you're starting or continuing beet breeding projects. You want to think about the purpose of your tasting or of your flavor evaluation. Do you want to really describe the flavors that that people are perceiving so that you can describe them on a website, on a seed packet, at the farmer's market? Do you care mostly that you're selecting something that people like, in which case a simple yes, no is most relevant? Are you curious about ways that the same cultivar performs in different preparations? So all valid things to do. You want to think about the method you use to prepare these beets. You probably want to pick something that is relevant to the way you actually eat them without applying too much extra flavor such that everything tastes the same as far as dressings or oils or salt. Some of that is okay as long as you do the same thing to everyone and you can still tell the differences between them. Consider your tasting environment. So it's a balance between the similarity of your tasting conditions to real life eating conditions versus controlling other variables that could influence how much you like or dislike that thing in terms of other things you're eating along with it, other people who are around whose opinions might influence you, all those things. And then finally, if you decide to collect data, think about what you're going to do with it before you collect it. And I'm happy to help with that if I can, if anybody has questions along those lines. And I put these pictures here. This was I think this is the cross that David and Mel used or close to it to produce the populations that the citizen seed trial is trialing. And this is the same cross I used for a genetic mapping project that I also did. So I think that's kind of cool. Oops. Just to say thank you to all the people and funders that made the work possible. And I'll stop sharing my screen so I can see you all.
Hello, yeah. Thanks so much for that, Solveig.
My pleasure.
I think that the next part of this, we're just going to go on to explain. Some of you are here because you're part of the Citizen Seed Trial, and you were growing out those beets that were a cross between a stripe and a gold. So we have both of the people who grew that original population on the call, myself and Mel Sylvestre from Grounded Acres Farm. And I'm just going to give you a brief description about what it is that you have and how it came to you. So the initial, you know what, I'm going to actually share my screen again and just put up a picture of those beets. And so this beet population that we started with, it came out of the Canovi variety trials in 2016. Both Mel and myself, Mel was growing them up at UBC Farm. There were, I believe, about 7 gold varieties and a few Chioggia varieties. And I think what Mel did was took the best of all of those gold and the best of all the Chioggias, and I got a population of them and she got a population of them. So multiple parents of each. My process was to, I planted the beets out, 1 stripe, 1 gold, 1 stripe, 1 gold down a row so that they would cross as much as possible. But I collected the seeds separately from the gold populations and from the stripe population. So one thing I noticed is the yield from the stripe population was at least twice what the gold was. So when I planted these 2 populations out again, I planted the population that came from the mother plants being Chioggias and the population that came from the mother plants being gold. And what you see in that photograph are those 2 populations. So the top picture is what the population that came out of the Chioggias looked like, and the bottom picture is what came out of the golds. So in order for me to select the next generation and know that I had crosses, it was really easy in the gold population to see what had crossed. So keep in mind, some of those golds in that population may have just been a cross with a gold and a gold. And in the top population, some of the Chioggias might be just a population cross of a Chioggia and a Chioggia. So we don't actually have a cross yet in that F1 generation. What I ended up doing was actually just taking a mixture of gold, stripes, reds, a wide diversity. As long as they were big and looked the right shape, I replanted them. In hindsight, I could have gone on to a much more controlled selection and probably planted just the stripes that came out of the gold population. And in doing so, I would have known that every plant that went into the next generation was an actual cross. But I didn't. So that wasn't my process. I just planted a mixture. I got a comment from Frank Morton, who has done some beet breeding and seed breeding. His comment was allow these to mix for 3 generations, throwing out only the undesirables, then begin to grow them as phenotypic groups, selecting the best for ongoing development. He also said you'll be done in a decade. And as we say, overnight success takes about 15 years. So that was my process for breeding those. And I'm going to actually share my screen one more time and introduce Mel Sylvestre. So she had her process. I'm just going to play a little video that she had put up online a little while ago about her process of this. Are you all seeing the screen?
I am Mel, Grounded Acre Organic Farm here.
Are you able to hear that?
And today I wanted to talk to you a little bit about a fun project that we have here that started a few years back actually, but we've kind of brought it with us. It's a breeding project. So what we have here is actually a cross between a golden beet and a Chioggia beets. So for those of you who may not be familiar, golden beets are kind of orangey yellow beets. Chioggia beets are the one that are striped, so pink and white. So when we cross both, what we got, it's kind of a stripy beets inside, but more of an orangey. And the main difference too is the foliage of the beets itself. It's still a green leaf, but we have more of a rib that's orange instead of red. Beets are what we call biennial plants, which means that they will produce their flowers and their seeds in their second years of life. So what we have here, those beets are actually got started last year from seeds, and we let them grow full size. We harvested them. We stored them over winter in a good cooler, and then in the spring, basically what we did is we actually took the beets, we did a little slice into it, we looked at the beet itself to be able to select what we really look for, because currently all of those beets are kind of mismatched. It's kind of having children not knowing what they're going to look like really. So we cut them out, we look what they're like, and if I like what I've seen, I replant them. And then this little beets currently here will become large plants with tons of flowers and seeds, and we'll collect those seeds, grow them again. And those are the seeds we're looking at here. And it does take a few years to get a good product and a new variety. So we're looking at probably, because they're biennials, another 6 years or so. We'll probably have a new variety of beets out there that we'll be able to sell. And in the meantime, even if it's not exactly what we're looking for, all the beets we're growing are still delicious, and you will be able to find them. Also, we grow them in our field currently, so this type of beet seeds and what you're seeing there, you'll be able to find them with Grounded Acre Organic this summer. So yeah, stay tuned to see what comes out of all this.
Hi, okay. Can you hear me again?
We can, David, yes.
I am now going to just pass it over to Mel. Mel, if there's anything you want to add about your process, or anything you want to add about this, and then we'll, after that, we'll open it up for questions.
Yeah, I just really don't like watching myself like that. So thanks for that. Can you hear me okay? Yeah. Okay. Yeah. So I guess, yeah, the process was, my process was similar to the one that David explain, I guess giving a little bit more background of how this started, because I found it like, it's part of the stories and it's an interesting part of it. So this whole breeding project, I was actually not very planned ahead of time. So not necessarily that I suggest doing that, but it is part of like the journey also as a farmer. Sometimes you come across things that you see and you're like, wait a minute, that's would be a great breeding project. You don't necessarily plan ahead. So just keep always your eyes out for exciting project like that. So this one, how it comes about is we were doing the BCC trial. Beets was one of the crop we were actually trialing just to try to figure out which one are the good golden beets out there and which one are the best Chioggia beets for our region, so that we could advise our seed producer on which one to grow for our farmers and for the market gardeners and gardeners. So we basically just trialed them out as a crop itself, not with the intention of crossing anything. And I got curious through the journey. We had all those like about 10 or a dozen different varieties that were commercial varieties that we were growing out. And when we came at the end of the season and we had all those beetroots available, we also got curious. We're like, well, if we're going to advise our seed producer to grow those varieties out for selling, it may be interesting to tell them what kind of yield they could expect from. And we knew already that golden yield very low seed yield compared to the Chioggia. But we were curious to see if there was actually major difference between the golden and major difference within the Chioggia in general, depending where they're from. So we grew them out to seed. That was kind of a little bit of a side project from the BCC trial. And being a side project on the working farm, we kind of dropped the ball a little bit on the data gathering of that part, but we do have anecdotal notes on that. So what ended up happening is that we saved the beetroot, and I should say like all that happened when I was at UBC Farm. So I was a wage worker, so I was allowing myself to do a little bit of those projects. Back in the day was a bit easier, giving myself the time to do those things, but still we were a production farm and not having like an over amount of time to take data. But anyway, we replanted those beets, we grew them out to seeds in varieties, but they were all, so compared to what David did, we wanted, because I kind of wanted to have an idea of yields, I wanted to keep them by variety to be able to identify them. So they were not planted as one Chioggia or one golden. So that's the first, in that first generation, David and I have actually like a different potential of crosses that happen. So once they got to seeds and the seeds were ready, then the next question was like, what the heck are we going to do with this? And that's when the breeding project kind of sparked, like the idea of doing that sparked, because before that, my only goal was to try to gather information about yield for those varieties. So it's like, well, we have those, may as well save them. We saved them by family again. So I gathered all seeds by family and replanted them the following year, a little bit as a progeny testing by, not family, I should say, but by variety still. But at this point, what we were expecting is that like there would be a few crosses. So we grew them out to observe what was happening. And similar to what David explained is like within the Chioggia population, I didn't see a lot of golden coming through. When we were cutting them open, we could have seen a little bit of difference, but I wasn't convinced that I had crossed, and it was very hard to tell from just looking at them if there was a proper cross. And there was a few golden, but I wasn't sure if it was us that just mixed it up when we were saving the seeds, and you know, seeds get stuck in cracks in places, and they get, because there was so little of them. I wasn't convinced. The one I was convinced about is like in the golden population that we had, like the 3 different, I think, golden variety that we replanted, we had not a large amount, but we had more that came out as Chioggia. And those I was a bit more convinced that they were an actual cross, just because of the number that was in it and all that. So we save mainly, that's the one we saved at UBC Farm. We saved from that golden, so that the mother plant was a golden, and then what we think is the pollen from Chioggia landed on those golden mother plants. So that's where the cross was. So we pursue with this. We grew those like the selected one, we grew them back out to seed again. That winter, our capacity to save the roots really well. Like, we didn't have a lot of roots basically that made it through, but we did have enough. And then we let them all cross, but we still saved those seeds out the following year. We saved the seeds by mother plants again. So we kept through our journey. Basically, we decided to let everything cross together in some ways, but always save the mother plants separate so we can test that as progeny later on. So it's like, in a way, we're kind of bulking everything up, but we're still controlling at least the maternal genetic of the plant. So that following year, like I say, we're like at year 4 now. We save seeds by mother plants. We grew it out again, and that was actually last year that we grew it out again. And then it became more obvious at that point that we actually did. I was happy because that's the point at which I was able to confirm, yes, we have a cross. Because what I was looking at in the field, it was a lot of like population or the variety or the mother plants. And sorry if I'm not using the right like language, but within like one lot, I would call it. I had Georgia, I had golden, and I had that cross that I was kind of looking for. Like I was explaining in the video, what I'm really after seeing it, what I really like was that striped beets inside a very like defined stripes, like what you find in Georgia, but with an orange background instead of like a white background. And I found that often those were associated with a very orange rib into the leaf and a nice green leaves. So that's kind of what I've never seen that sold as a commercial beets. I'm sure that some other people might have done it somewhere, and they may be available in some obscure seed company that I'm not aware of. But commercially, it's not something you find. And I got into it. And I should also admit, I'm a huge fan of beets to start with. So that's why I got a hook on that breeding project, because there's this other breeding project that came my way that I just dropped because I was just not passionate enough about. But yeah, so that year, last year, we basically saved the roots again. We store those roots. That's the winter that I moved out of UBC Farms. So I didn't get to do the selection, but I'm glad to, if I'm correct in a way, I think Solveig, you participated in that selection of fruits with one of my colleagues from UBC Farm that took over event. They did the selection, and then I actually just drove to Vancouver one day and grabbed a bunch of roots and brought them to my new farm. So I was able to plant that. So at this stage, I would say we have UBC Farm and ourselves at Grounded Acres. We have the same population, but what's going to be interesting moving forward is to know that David has its own population. He's selecting for something, and we may be selecting for the same thing. Who knows? I'll have my own, and then UBC Farm may eventually also have their own. So we may even have like 3 different varieties coming out of this selection. They may be the same. I would be curious to grow them side by side. It's something I think we should be doing in future years, is to grow what we've saved so far and see if we have selected for differences or just bulk everything together. But yeah, I think it's, sorry. It's and I should say also last year when I grew them out in the field to observe if there was differences, I did 2 replicates of each because I didn't have a lot of space and I only had 1 bed and 1 row, and I knew that field was variable. I just didn't want to throw something out based on the fact that maybe the edge of the field wasn't as productive as the other. Because at this point, I should admit too, I'm not selecting with flavor in mind. I do want to add flavor to my selection process, but I kind of want to narrow down that visual aspect of the beet that I'm looking for before I start selecting for flavor. So I'm not writing off flavors. I think it's a huge and important part of what we're doing, but I know as a market gardener, shopper shop with their eyes more than with their mouth. And with beets, I don't think our population, human population around as of a developed palates for beets, beets. And because we started also with varieties, common commercial varieties, I kind of trust that what we throw in the lots at the beginning are all pretty good beets to start with. So I'm not expecting something going completely off, but I could be wrong there again. But yeah, we would definitely, before I release anything in a way as like a commercial variety, I would definitely make sure that the flavor is good. But we're probably talking, like I said, in the video, or probably talking about another 3 generation before I can get comfortable. But the interesting part, as a lot of you know, is that after I grew it out in that second seed generation, after we grew it out, we realized, well, all of those beets are great. They're good, and it's just a mix. So may as well, we have a lot of seeds. When we replant for progeny testing, we maybe use like what, 1% of the seeds of a plant, and then we're stuck with that 99% of like a big bag of beet seeds. And I was like, that's a waste. Those are all beets. And that's when I think David and I had the same idea without even talking to each other. Is that like, well, let's just sell that beet lot and just describe and tell people what they're going to find in there. And this is what we grew even on our farm this year. People at the market love it. We call them rainbow beets. And people love them even more than our red bunch. So it's actually kind of nice to have. We may even keep rainbow beets as like a part of our like varieties that we offer through the co-op, at least until we have that new varieties available. But yeah, so that's kind of what we're at right now. They're growing up to seeds right now. What I have out there, like you saw in the video, now the plants are very large. Have a very healthy population of rats living close by. So I'm hoping I'm going to be able to get some seeds out of them. I had to stake them up because of it. But anyway, I'm pretty sure I'll get some seeds. So my next step now will be I will collect the seeds again from all of those individual plants. I have about 30 plants right now. And I'll grow them out next year, and I'll do the same process as I talked about in the video of like progeny testing again from the mother plants, selecting out of that and keep moving on with that. But yeah, and we'll keep making those seeds available through the co-op at least. Yeah, I think that's about it. What I wanted to share. See if there's any question about it. Shoot.
Okay, well, happy to move on to a question and answer period. So I don't know, Rebecca, if there's anything in the chat that's come up, but if anybody has a question, feel free to either throw it in the chat or unmute yourself and ask.
I have passed on to Solveig 3 questions that came up during her presentation. Maybe she can just speak to those.
Sure, let's see. Where can we find my thesis? If you happen to have university access to the ProQuest dissertation system, you can get it that way, just under Google Silvia Hansen or search for Silvia Hansen Beets. If you don't, send me an email. I'll put my email in the chat and I'll just send you the PDF. I don't think it's publicly viewable. I've submitted that article on the participatory beet breeding program to a journal, and it's being reviewed, and one of these days it'll be published, but not yet. How many seasons has the study been going on? So the first year of selection was 2016. The starting populations were made in the 3 previous seasons. So I guess starting in 2012 or 13. And then we did 2 years of selection for like to get the base phenotypes, and then another 2 years of selection while we were doing the tastings. So like once we knew what we were looking for in terms of like farm performance and color and stuff, we just kept refining that in order to like choose which families to keep. And we used the tasting to characterize flavor and see how much people liked that. I just want to say, I think Mel's using a really logical strategy for something that comes from 2 marketable or 2 commercial cultivars that are widely acted. Like the range of flavor compounds and the variability you're likely to see between them, between families, is pretty low. So I think you're totally right that you're going to end up with something that's like pretty acceptable. And like, so getting the color you want is like, that's what's going to make that population distinctive. So I think that's really smart. And then if I can clarify where the study was done more specifically in terms of region. So the outreach beets were bred at the West Madison Agricultural Station outside of Madison, Wisconsin. It's about a 15-minute drive from downtown Madison. And then the farm, TP Produce, is like 40 minutes south of Madison on a bit sandier soil, like a sandy loam. The research station has a silt clay loam soil, both organically managed. Does that get to those questions? Feel free to speak up.
Sounds wonderful. I do have another question, perhaps for all of you. It's how much does soil or nutrients and seasonal weather affect flavor? Rob has been growing mangos, which have strong metallic flavor, not in all individuals, but he's curious as to whether these are coming from a trait or the soil and weather.
So in my humble opinion, one word that some chefs actually gave me after we made the flavor lexicon to describe jasmine flavor was sharp. That he could be tasting jasmine, and in mangoes especially. I guess I don't know about jasmine levels in mangoes. In sugar beet, jasmine's very high, so it could be that it's a high jasmine variety, and that's how the jasmine is being perceived. But the real answer is I'm not sure, except that the more stressful conditions are, the more secondary metabolites or just compounds that plants produce to protect themselves are produced, and those often have strong flavors. So yeah, it makes total sense that in more stressful conditions you would get beets with stronger, harsher flavors.
And also, if folks are curious, like the variety trial that was done, I think it's 2016, that was trialing all those commercial varieties. Like flavor was a huge part, I think, of our evaluation. But what was really interesting is we had all the beets grown at UBC Farm as the mother site, but we had about, I think, 5 or 6 different farms across the province growing them out to as sister or baby site. And there's a report available on that. I think it's available on the BC Seeds, Farm Folks, Seedy Folks website. And it was really interesting to me because the one that I liked the best in the way at UBC, or even in terms of agronomic traits, that they were like yielding higher or looking better, were different than even folks that I know personally, that and a farm that I worked at on Vancouver Island had a complete different take on it. Like they really like another one varieties that mine didn't do well there. So, I mean, I can speak for flavor. Like I said, like I think Solveig is really better placed and speak of how that would influence flavor, but agronomic traits gets influenced by the type of soil and condition you have for sure for beets. And pretty much every plant. That's why the word terroir come from. Really, it's like the type of soil you have will influence the plants, and therefore flavors and everything that comes along with it. So, yeah, I think for folks of you, like those of you that are into beets, like I think looking at that variety trial results that was published by Alexandria Lyons, could be an interesting first steps for sure.
I'll paste a link to I know it's on the BC Food web page. I'll post a link to that in the chat and people can find the bee report. Yeah.
I have a question from Liv here asking, stating first, so interesting to hear about the flow of seed selection, Mel. Can we hear more about how traits are brought out? Is the order usually something like 1, appearance, 2, flavor, 3, germination somewhere? ?
Yeah, that's actually like part of, I guess I would say it's a very personal question that I don't want to share, but I think that's what would be interesting between David and myself and maybe Evan at UBC Farm now. We may all have a list of selection of traits that we're looking at, and they may be in different orders. I think as a grower myself, I'm not just a seed producer, I'm a grower. So I do care a lot about appearance because that's how our shopper like work. They look at the beets first. So I think that's why for me, obviously that's a huge part of it. But I think along the road, as a grower, like if I notice that one lot that I'm carrying always germinate badly, I think it will be a no-brainer that I would probably eliminate that one eventually. But to be honest, because this is not a breeding project I launched in with an idea in mind and then I wanted to see it happen, it's kind of like it's sticking to me more than anything else because I like it. I'm not having a very precise idea at this point. Like I think I'm more focusing on the visual right now of the color, but any other agronomic traits will matter along the way. The good thing actually I like to share about this is, I mean, I think if any of you have grown Chioggia and golden and then red beets, like open pollinated red beets, you may have noticed in general, golden genetics are pretty weak. And from my understood, and so if you can correct me on that, is that like they kind of got dropped off the map at one point, and certain things people were just not interested in golden. So the genetics that may have been out there 100 years ago kind of got lost. So we're stuck with a very weak genetics, I think, with the golden beets that we have to work with. There's not a lot of different genetic out there. So that's what I found was interesting in crossing the two, is to like get the vigor. And if anybody have grown Chioggia, it's by far like the most vigorous beets, the best germination, the best seal, the best foliage. They just are very, and I don't know if it's from that they originally have some hybrid vigor still in them or anything like that, but by crossing the two in a way, I've noticed a lot of hybrid vigor and trying to bring back that kind of golden color on the table with the agronomic trait that we like as farmer, which is high yields and like, you know, and good germination. It's very exciting too, because I think as part of the BCC trial, when we were trialing, I was kind of hoping to find an obscure golden beet out there that would perform better than what we're used to. Like nothing against Touchstone Gold or even Bulldor. Like they're okay, or Sunrise, I think was another one. But they're still like overall in the big picture, they're still never perform as well as like even a red beet, and surely not as well as a Chioggia beet. But unfortunately, some chef, well, not unfortunately, but some chef and some customers want that golden beet and don't want the Chioggia. So as farmers, I know that I would prefer having higher yield when I grow golden, but I will still grow them because my customer wants them. But I'm not making as much money by square feet in growing them. And I'm still weeding them as much as the other one, if it's not even more, because their yield are not as good.
Thanks, Solveig, for putting the in the chat. If anybody's interested, there's the results of that 2016 beet trial, so you can see all the varieties that we were looking at. I just want to make a quick comment because Mel alluded to this as a farmer trying to do a breeding project off the side of your field. It's not very, it's challenging. You don't get paid for that work, and farmers aren't making a lot of money. So we tend to not put a lot of effort into it. What I've seen, the kind of work that can be supported through some of the programs like participatory plant breeding programs, is a lot of that work being taken off the farmer's plate. I might have done this initial breeding project quite differently if I had some support to do it. And the other thing I'll mention, because Mel alluded to it as well, is that both of these varieties are actually up for sale on the BC EcoSeed website. So you can get them even though they're not finished varieties. And you know, when I first started growing seeds and thinking about breeding, I thought, yeah, it's going to take me 10 years before I have a variety I can offer. And during those 10 years, you're throwing out all of the work that you put into them. As Mel said, you use a tiny amount of seed and then you're going to throw away the rest, and nobody's paying you for that work. So in some ways, going and buying seeds from an existing and ongoing breeding project is a way to support that ongoing work. That way, at the end of 10 years, when we have a finished variety, we don't need to patent it to protect all of our not that we would, but you know, intellectual property rights and the work that went into it, we've actually just been able to pay ourselves all along the way. So in some ways it wasn't a deliberate model, but we both decided to do it at the same time, and it seems to be working. And my observation as well is that rows of mixed beets, I'm getting great results. It's a great yield and really easy to harvest from.
Yeah, that's a really cool model, and it's great that it's providing some like financial support for the breeding process, because obviously at universities, you know, there's all this infrastructure intentionally, because it takes infrastructure to breed crops. You can't do it for free. But yeah, it's a cool alternative.
I don't have any further questions here in the chat. I did have one personal question that I would love to know if any of you know what are some of the dominant traits in the beets. I know Solveig may have read about this.
I mean, I read up on beet color genetics this morning because I anticipated a question something like this. Are you thinking about dominant traits for color or other traits?
Yeah, well, I was thinking about listening to Mel and thinking about which ones are not showing up less in the population. And is it just because things weren't getting crossed, or is it because, you know, like when I save seeds from some of my flower populations, I'll get like all red if I don't select properly.
Totally. I'm going to see if I can copy and paste this, see if it'll work. Yeah. Okay. So there are two loci, genetic loci in beet that control color, called the R and the Y locus for red and yellow. Basically, the R locus, when it's a functional gene, when it codes for the enzyme it's supposed to code for, it here, I'm gonna put it up on my screen so I say it right. Not that anybody's grading me, but okay. It takes the precursor molecule, which is an amino acid called tyrosine, which is one of it's in plants, and it makes let's see. Well, it makes one compound. To get red, you have to have that compound plus beta-lamic acid. The Y gene, the functional Y gene, makes that beta-lamic acid. So if you just have that, you get yellow beets. So if the R locus is recessive and there's nothing to mix with that beta-lamic acid, you just get the yellow pigments, which are beta-xanthins. If you do have both that beta-lamic acid plus the compound that the R gene makes, they mix and you get red. If you don't have the beta-lamic acid from the Y gene, if you have the recessive Y gene, you can have all of the called cyclodopa, the thing that the red gene makes. You can have all that you want, but if it doesn't mix with what the Y gene makes, you don't get any pigment at all. You get white beets. So does that help? So let's see. So you're crossing a yellow beet, which is recessive R, at least one dominant Y gene, with a Chioggia beet, which is in the red family. Like if there's any red pigment, it's coming from that mixture of those two compounds. So at least in the first year, it totally makes sense. You're getting a dominant R gene from every Chioggia beet, so it's going to cover up the recessive R genes in the yellow beet. And the few yellows you did get breaking through are probably because that red, the red genotype in the Chioggia was like that, you know? So there was a recessive R gene, or like it was heterozygous for the R gene in the Chioggia, and you cross that to yellow and you get a few yellows coming out, but mostly reds. Does that make sense?
That's very cool. It makes sense to me.
Right?
I have a question from Liz asking, so it would be impossible to, for example, have a white, yellow, red striped beet?
So I haven't seen that. I have seen the alternation between golden and red, like that you're all getting in some of your beets. My professor at the UW also has some that look like that. And I've seen like zoned yellow beets, so alternation between yellow and white, which I think is just that's a function of the amount of pigment that's produced. Just like Chioggia is a function of like low pigment level, so it's only distributed like the rings of beets like are cambial layers. So, and cambia is like the growth tissues that makes the roots swell. So you have like all these layers of cambia that swell, and you have like just like, well, parenchyma, but like just tissue between them. So if there's only a little pigment, it's just distributed along those along the cambial rings. Anyway, never seen red, yellow, white. That'd be super cool, but I'm not I'm not sure how you pull that off. But you know, it would sure be incredible.
And I noticed there was a question there. Somebody asked, have we ever thought of mixing Swiss chard into the mix? And as somebody that's taken on a project that I already know is going to be about 10 years, I think
Right.
I think Swiss chard would probably turn it into a 20-year project if you actually want beets out of it. If you're, you know, harvesting for the greens, that might be different. That would be my sense of it. I've certainly thought of turning this into extending this project and mixing in some cylindrica beets, because all of the ones we selected for were round, but even that is gonna add multiple years of selection to the project, which is no reason not to do it. So we'll see, maybe next year. But any other comments about mixing chard in?
Yeah, I would probably stay away from it personally, because the good thing about chard is it's been selected for its foliage and a lot of foliage. And I found with Chioggia in the lot already, you get a lot of foliage out of those beets. And if you think about processing as a farmer, no, it's great to have a lot of foliage, but it also makes it a mess to harvest and clean up the beets and bunch it and all that. So I would not want to have more foliar growth on those beets than what I got right now, but it could be a nice thing or fun thing to do for sure. And also, like, I think it's what's fun about, like, you know, you were saying like adding like potentially another 12 years on the breeding project is that there's nothing that stops us to have multiple lines going at the same time too. Like, I mean, that's one thing I've been expecting that could come up along the road is like, maybe at one point when I select, I may just be like, oh, I love both of you. Like, which are different, like, you know, they look different, and I may just take on like two different line, which makes it complicated in the way of being on a small farm. I would probably have to find myself like a farmer friend I would want to grow under the line at the same time, otherwise they would cross, and if I want to keep them separate. And right now, infrastructure wise, I don't want to have to start covering up beets or anything like that. Like, I like having only one of that species growing at the same time. But yeah, I'm going to try to probably stop myself from doing that, because I already have about a dozen bean breeding project going on, and leeks and tomatillos. And yeah, like David said, that doesn't really pay the bill. So I'm sure my partner will tell me not to do 2 lines of beets, but we'll see.
Yeah, we did some experimental beet chard crosses at the university, and it works. You get plants, but it's yeah, I guess you'd have to just think about what you like. If you want a full-size beet root and also the chard top, just got to think about how, like, that could potentially be like a dual function crop. But I think like there would definitely be a lot of trade-offs, because chard has been definitely bred for the wide petioles and big leaves, and beet not. But
there are no further questions here in the chat, but I'll pass it back to you, David.
Okay, well, this is probably good timing then. We're sort of 5 minutes from the end. We do have, we would love if you would share some information with us just before we leave. So I believe Rebecca is going to share a poll. I'm just going to put up share my screen one more time. Okay. Are you seeing my screen? Okay.
We see your back.
The whole
backdrop.
thing.
Yeah, we don't see the presentation.
Okay, let me just stop that again. Let's try that again. How about now? Do you see it now?
We see it now.
Okay, so yeah, this is just the closing. So yeah, do people see, hopefully you see the poll that's just come up. There's just a couple quick poll responses. You're also absolutely welcome to add any information or any feedback into the chat, or email us separately for sure. So this is the feedback we're looking for, just your change in knowledge and sort of overall quality of the event. Feel free to send any additional feedback to bcseeds@farmfolkcityfolk.ca. And stay connected. So these are a couple sites, seedsecurity.ca. The next webinar, I think, will be breeding and selecting specialty varieties sometime in September. But we have a whole series of webinars coming up between now and the end of the year. So check them out, and would love to see you there. And yeah, thanks everybody for coming. We'll see you next webinar.